while we're getting this going, I'd like to say thank you for inviting me. And when you hear the word interdisciplinarity, it means different things to different people. For example, when I just heard Francois Jacob talk about on the 50th anniversary discovery of the lac operon, there were people working on episomes, plasmids, bacteriophages, and really it was a combination, also lactose metabolism that came together, and really that was what led to the discovery of the lac operon over 50 years ago. And I've been very fortunate. The way I've been brought up is really, I've been exposed to interdisciplinarity pretty much the entire time of my science career. So when I was doing my PhD in a biochemistry department, I was actually working on microbiology. I then did a postdoc in a microbiology lab where I studied cell biology. I then was recruited to a new group that just formed, this is back in the 1980s, where they took engineers, people working on plants, and people working on animals and humans, threw us all together, really much like you're doing here now, but that was 20-odd years ago, and it was very radical at the time. And so then I became a member of the Peter Wall Institute of Advanced Studies, which has a partnership with the college. And now interdisciplinarity means me talking to geographers, historians, philosophers, which is really very different than a cell biologist talking to, say, an immunologist. So this is the 17th talk today. My guess is most of you are completely brain dead, despite numerous talks on neurons, etc. So what I'm going to do today is I'm going to ease off on the data. I'm doing other talks later on, so there'll be more data there. And I want to tell you the story of all the different approaches we've used to really try and understand a particular problem. Because I think the reason we choose interdisciplinarity is it gives us different approaches to a particular problem. And I'm fond of saying, if this is the problem, it looks like this to a molecular biologist, this to an immunologist, this to a physician, this to a medical microbiologist. It's the same problem, but they see it very differently. And over the years, I've tried to form my lab in that way, where I recruit people. I really like taking people in my lab that don't know anything about what we actually work on, because I find they bring a very unique perspective to the thing. So the problem is basically diarrhea. I will warn you right now, this is the problem, and at the end of this seminar, you will know much more about diarrhea. Don't worry, it's not too interesting. And I'm going to talk about an agent that causes this. That is what we call pathogenic E. coli. So there's two kinds of, there's actually 7 different kinds of E. coli that cause disease in people. So most of you know E. coli is a lab strain that's been very well studied, probably the best studied microbe on this planet. But there's actually species or types of E. coli that cause disease in people, and they can cause diarrhea. 5 of them cause diarrhea. 1 causes meningitis, and the other one causes urinary tract infection. So I'm going to talk about 2 that basically cause diarrhea. These 2 are very similar, so they'll basically be kind of merged together. So the first one is something called enteropathogenic E. coli, or EPIC. And what this does is causes severe diarrhea in young children, less than a year of age, especially in developing countries. We don't see it so much in our societies anymore, but in developing countries, this plays a huge problem in childhood health. And what this bacterium does is rather quite remarkable, that when these children swallow the organism, it goes down into the intestine and actually adheres to the intestinal wall and forms this, we call this a pedestal structure underneath it. So unlike the Shigella talk you've heard about that Philippe and Guy talked about yesterday, this bacterium actually resides on the intestinal cell surface and basically reprograms the cell, so it's then got this throne or this pedestal upon which it adheres. The other one that's a very close relative of this is something called enterohemorrhagic E. coli. And in North America, this is a big issue when you eat poorly cooked hamburgers. Now, I know in France, when you order a hamburger, it comes raw, and that's okay. That's because you don't have this particular E. coli here. I actually sent mine back the other day to get it well cooked at the Equire. I just couldn't deal with a raw hamburger here. But in North America, never order a raw hamburger, because this organism is there. And the way it comes about is basically cows carry this organism in them, and they secrete this organism in the environment. And it's this contamination of either hamburgers due to fecal contamination that causes it, or the cows defecate and then goes into water, and then it can lead to waterborne outbreaks. So it's got an LPS serotype of something called O157, so sometimes it's called O157. So those are 2 E. coli that cause diarrhea in people, and that basically is our problem. So what I want to spend the next bit of time discussing is, okay, that's a problem. How do we learn about it, understand it, and then ultimately do something about it? Because that's where I think we needed to go. So first of all, you have to have a system, a model system to study disease. And there are some model systems with EPEC, but they're in humans, but they're obviously very difficult models to use. So we resort to a bacterium called Citrobacter rodentium. It's unfortunate this isn't called E. coli. This is basically pathogenic E. coli in mice. It's a lovely mouse model, and it's actually a natural pathogen of mice. So mice get this thing, and they actually get real disease. So this is not an artificial model. It's a natural infections of mice. And it does all the same things as the other E. coli. It basically forms those pedestals under these things. And it also causes proliferation of the intestinal cells, colonic hyperplasia, so that 10 days after infection, the intestine in the mouse goes from here to extreme proliferation to here. And this is basically our mouse model of this disease. Okay, so we have this bacterium. It causes disease. How do we go about figuring out what it does? Well, many years ago, we and others basically saturated the chromosome with transposon mutations, and we found that all the mutations that seem to affect the formation of that pedestal all clustered in one area on the chromosome, less than 1% of the chromosome, and it's something called a pathogenicity island. Basically, if you're taking the E. coli genome sequence, you come along E. coli K12, then you get to this very different sequence here, and then 35 kilobases later, you then go back into the E. coli. It's actually a tRNA gene. So this is plunked in from somewhere. We don't know where, but basically this gene cassette is dropped into here, and we started to figure out that this is where most of the genes were that caused the problems, or the disease. When we started to figure out more, what we learned is that it encoded a type 3 secretion system. Now, this has already been talked about twice at this meeting. I'll say more about this. This is a system the bacteria have to make basically a hypodermic syringe to drive bacterial molecules into host cells. And that way they can deliver a whole bunch of things into cells and then reprogram the cell. And we saw it with Shigella and the actual invasion, and I'll talk more about it here. Then, at the other end of this region are proteins that are basically secreted by the type 3 system, and these are what basically go into the host cell or form the mechanism to drive the protein into the host cell. And in between, if you made mutations in here, it seemed to block the really tight, intimate adherence to these things, and I'll say more about that in a moment. Okay, so we got some genes that are involved in doing this. What do we do next? Well, over the years, spent a lot of time studying this thing, and what we learned is that this type 3 secretion is really many proteins that basically span the inner and the outer membranes of the bacterium. So there's about 20 proteins in here that basically form this hypodermic syringe. It has an ATPase that then drives this whole process, so here's where the energy comes from. And then the molecules that are going to get secreted basically have these chaperones that bind them in the cytosol of the bacteria, deliver them to the system, and then they drive them out through this system into the host cell. So we started to do some structural analysis of these things in collaboration with Natalie Strank at our university, and she's a crystallography lab. And so pretty much we went through many of these proteins here to actually figure out what was going on. One of the things that we soon realized is there's a big pore in here. There's these donut-shaped structures of this particular protein here in the inner membrane, and basically there's a hollow tube that allows the bacteria to secrete proteins out of here. So we started to solve these structures, and we solved many structures. I'm not going to go through these, and over the years we've published many of these structures, but basically we've got a much better structural understanding now and at a molecular level of what this type 3 base looks like that's embedded in this membrane. And we've solved many, many structures over the years, many of which aren't up here. But so we're really starting to figure out at a molecular level how this molecular syringe actually works. Well, the syringe pokes into mammalian cells. And we wanted to have a look at what this tip of the needle, shall we say. So if you think of this as a needle, here's the barrel of the syringe here, here's the needle of the syringe here, and here's the very tip here. And we know what these proteins are. So we started to have a look at this, and this thing, protein that makes up this needle of the syringe is something called ESPA. So we had a look at that, and then again with Natalie's lab, we basically used cryo-electron microscopy to have a look at what these needles looked like, and we were then able to figure out generally how this thing actually looks like and how it actually then goes up to the mammalian cell. Okay, so we've got a lot of structural data on how this type 3 system is. I've told you it's used to deliver bacterial molecules into the cell. So what does it deliver? What are those molecules? And that's a major focus of this field, because obviously this is what's going to say you then cause disease, and how do you cause disease. So over the years we studied this and started to figure things out. We identified what the needle of the syringe was. We identified what the two proteins were that made the pore in the mammalian membrane. So this is the epithelial cell here. And then we started to find molecules that are driven into the mammalian cell. These are things called effectors because they have an effect on the host cell. And you can see that several of them are encoded with this region, and if you scan through this list, you can see they're involved in cytoskeletal rearrangements, etc. And again, I don't want to go into these in any detail, but I do want to talk to you about one, this thing called TEER. This is a bacterial protein that starts out in the cytoplasm of bacteria, gets shoveled down into the mammalian cell, and ends up in the mammalian membrane. It becomes a transmembrane protein in the mammalian membrane. Now, not only does it lodge itself there, it then binds to a protein that's on the bacterial surface, this thing called intimin. And this is what these two proteins together are what give you the intimin adherence, locking the bacterium down. So here a bacterium actually injected its own receptor into the intestinal cells, to which it then adheres. Now, TEER doesn't only do that. The other end of TEER actually gets tyrosine phosphorylated and nucleates actin that actually then builds that pedestal underneath it. So an amazingly complex delivered system for a bacterium to basically stick to an intestinal epithelial cell. So we worked on this protein quite a bit over the years, and we learned a lot about it. And basically, I've already told you it's a transmembrane protein here. We solved the structure of it, complex with intimin, and so we now know at a molecular detail how these proteins actually bind, and we know right down to the residue level which residues are key to allow this bacterium to bind to the host cell. Okay, so we've got lots of information there, and we're starting to get along in terms that works. Well, how do you form this pedestal, or this big protrusion you see in epithelial cells? Well, this was probably about a 10-year studies in our lab over the years trying to figure out all this. And really this led us into the whole world of cytoskeleton. And if there's anybody that are cytoskeletal jocks and jockettes in the audience, you'll recognize many of these proteins here. Basically, this is an actin-rich structure that the bacteria triggers. It uses these proteins in white, and again, I'm not going to go to the details of this, but we now understand how this actually does it, to basically polymerize at the tip actin, which then forms the pedestal under these things. So it reprograms the mammalian cytoskeleton to build this pedestal. Now, if you're with me so far, you're saying, why the heck does the bacterium go to all this trouble to build this great big pedestal? We don't have a good answer. We know that this is key for the process, and if you knock out such as TEER and things like this, then they don't happen, the bacteria doesn't cause disease. But we really don't know. We think it's probably to prevent the bacterium from being internalized and actually to build a structure to live on the cell surface. So we figured all this out, and we did the molecular Lego and identified it. And again, I'm just kind of a summary of many years of work that basically the TEER molecule gets tyrosine phosphorylate and binds an SH2 protein that then grabs this actin polymerization machinery that then builds the pedestal under it. So a terrifically complex process for bacterium just to stick to the intestinal epithelial cell to then try and trigger diarrhea. Okay, so let's go back, and I've told you there was this big genetic chunk in this region here, and this is where we knew we had to look for all the answers. And we were picking off proteins one by one, trying to figure what they did. And then we said, well, why don't we just knock each of them out? There's only 41 open reading frames in there. And at the time, we came from the school of one gene, one protein, one PhD student. And okay, well, there's 41. That's going to be an awful lot of students. But luckily, technology has advanced. This is no longer a big deal. And we were able to do this in a few months. So the lab went through and knocked out each one of these things. And then we started to screen, well, what's the effect? Because before we started, we only knew the function of 8 of these proteins, and the other, whatever 41 minus 8 is, remained unknown. So then we screened them for various phenotypes and asked what happened. Well, this is one of those experiments that worked extremely well. We basically got a function for every one of those proteins based on the screens we had in the lab. So we identified new positive and negative regulators. We now know there's 23 genes that are needed to form that type 3 apparatus. You need 32 to make the pedestal. The neat thing is, every single one of these was needed to cause full disease. So obviously this is a real virulence cassette, and it's really needed. But then the plot thickens, as it always does in science. And we knew there were a bunch of effects that were triggered by this bacterium, but we couldn't blame them on some of the genes in there. And that told us there was more. There was more proteins involved in this whole thing. So we used basically proteomics, et cetera, to then identify what some of the other secreted proteins were. And the bottom line is we started to find other effectors. These are molecules going to the host cell, scattered in little islands all over the chromosome. And we started to find a whole bunch of these things. And generally in E. coli species now, there's probably about between 30 and 40 in each of the 3 EPEC, EHEC, and Citrobacter I've now told you about. So there's all these other things that are scattered around the chromosome. They're doing something as well as that genetic chunk that makes the pedestal. So obviously our life just got a lot more complex. So we have 30, 40 proteins that are injected into host cells to cause disease. How do we figure out what they're doing? And this is an area that the entire field of molecular pathogenesis is focusing on. Many different gram-negative bacteria have these type 3 systems, and really the current state of the art is, well, what are the effectors? What are they doing in the host cell? So this is where we sort of took a systems approach to it, and we basically used the differential labeling system and then proteomics using labeled and unlabeled cells to find out what was binding to each one of these effectors. So we took each one of these effectors and we either expressed it in mammalian cells and then pulled it out to see what it was pulling out of the mammalian cells, or if they wouldn't express mammalian cells, we cloned them into E. coli, expressed them there, and used these as an affinity column, and basically tried to identify the binding partners. This met with pretty good success. We basically identified all the ones we're known. So here's a list of all the type 3 effectors that are injected into host cells, and the ones in red are proteins we've now identified that they actually interact with. But it didn't get us them all, obviously. And there's many reasons why we didn't find them. They might be enzymatic and transient. They might need one effector to go in to do something before we can see the other effect. But anyway, this continues to be a work in progress as we pick through this list and identify all the binding partners. Okay, so that's good. That works in tissue culture, but we work on disease. So let's go back to disease models and try and figure out how do we actually understand the biology of causing diarrhea. So we returned to our mouse model, or our mouse infection model, and we started to probe what goes in there. One of the things we wanted to do was really convince ourselves that we were on the right track from the work we got in the cultured epithelial cells. And so first things we did was to have a look to see if TIR, that molecule goes in the host cell, is actually under the bacterium in the intestine of infected animals. And you can see the green bacteria here and the red TIR underneath them, just like we would have predicted and just like we see in epithelial cells. Okay, that's good. That means we're on the right track in terms of the disease models. Now, I've been focusing on the bacterium, but when you think about host-pathogen interactions, you got to think about the host too. And we spent a lot of time doing this, and this is just a various knockout animals in T and B cell type responses. And I don't want to go through the details again, but just bottom line is, if the T cell mediate the hyperplasia, the proliferation, and you need both B and T cells needed for bacterial clearance. And we spent probably the last 8 to 10 years going through many, many knockouts in different innate pathways, really studying the role of the host contributing to this, and finding all sorts of components. For example, who would have guessed that T cells actually mediate this proliferation of the intestinal epithelial cells? So there's the host components to that. Okay, I told you this thing causes diarrhea. Well, how do you look at diarrhea in these animals? So these are experiments where basically Julian Gutman, who was a postdoc in the lab, he took sometimes simplicity is beautiful in science. He said, okay, if they're causing diarrhea, they're probably breaking the gut barriers. And if we put in, say, a biotin-labeled substrate and lumen in the gut, if the normal gut, it's impermeable, it's not going to diffuse through. If we infect with Citrobacter, you're going to break the barriers and you're going to see the diffusion of the label going through. And that's exactly what Julian saw. Okay, so now we have a model to actually look at gut permeability and barriers. And we went through all the mutants we generated. I don't want to go into the details here, but basically found a single effector that seems to be critical for busting the tight junctions and the junctional structures in these things to allow this permeability of the gut barrier. And if you knock that protein out, you can see that you no longer break the barrier, whereas the wild type and another mutant still break this barrier. So here we have a component that seems to be involved in basically disrupting the intestinal epithelial integrity. Well, how do you measure diarrhea? Again, Julian took a very simple approach. He said, well, heck, I'll just take the feces of these animals, weigh it, and then wait a bit, let it dry off, and get rid of the water content and weigh it again. And what you probably didn't know is that anything over 80% water content is rated as diarrhea in these animals. So by doing that, he basically showed that the wild type causes diarrhea. You can see the wild type causing diarrhea here. A type 3 mutation doesn't. And again, that one effector I told you about, it causes a much decreased amount in the diarrhea. So again, confirming the idea that the busting of the junctional integrity is critical for causing the fluid accumulation. And you can see, so here's an unaffected versus a wild type. This is very fluidy, etc. Okay, so I've talked a bit about the bacteria, I've talked a bit about the host. I now want to talk a little bit about the microbiota, the normal flora that's in the gut. And one of the things I do with my lab is we go on what I call retreats about every year and a half. We live in Vancouver, very pretty place, and we always take these nice places, such as Whistler, which is a ski resort, or one of these small islands there. And we have these really brainstorming sessions where we try and get people outside the envelope and really think about other things we should be studying. Really, get away from the bench, because the bench, you never think what you're doing more than a week ahead, whereas in science you should be thinking the big picture. And, of course, a lot of alcohol helps at these sessions. So one of the sessions a few years ago was this question came up. What happens to the microbiota during diarrheal disease? Easy question, right? I mean, microbiologists have been studying this forever, and they must know what goes on. And so when we came back to the lab and started to probe this question, we really realized the question had not been answered. It was answered for cholera, which is a severe diarrhea, and that's when it basically turns into a monoculture of the Vibrio cholerae organism. But all the other diarrheas, nobody had really looked. So we then wanted to see what happens to this. So we turned to our mouse model again, which I've told you is a natural infection model. And if you give the mice Citrobacter, the numbers of the Citrobacter increase up until about 7 days when the infection really starts. And days 7 to 14 is maximal infection. And then the TMB cells come in and they clear the organism. This is a natural infection. You get sick, you have symptoms, you get better. So here we have a 28 days period to have a look at what's happens to the microbiota during these infection phases. So this is what I'm sure you've all wanted to know. This is what the microbiota looks like of a C57 black 6 feces. So we basically are stating the bacteria, and it's very pretty. They're different shapes and sizes, and this is what normal flora looks like in the C57 black 6 mouse. What happens when you infect? Well, what do you think would happen? I assume that, first of all, the microbiota are all gone. I mean, they're going to have diarrhea, and it's going to be only Citrobacter, because I only care about the pathogen, and that's what it must be. And so I assumed that would occur, and of course I assumed wrong. So I'm just going to show you, this is before, and then the next slide is after. So there's before, there's after. So before, you've got lots of different shapes and things. You go after, what do you see? Well, you see a decrease in the number of bacteria. Okay, they went down about 90% or so. That makes sense. But what you also see is a shift in the distribution of basically the small round ones versus these cones. There's been a shift in the composition of these things. Now, the other thing is this is labeled for Citrobacter. This is 7 days in the infection. It's maximal diarrhea, and we labeled the bacterium red. Where's the red? Well, if you look closely, you might not be able to see, but there's one there, one there. There's only 2 Citrobacter here, even though there's extreme diarrhea at this time. So, okay, it doesn't take over the thing. It obviously comes up, and you can see it, but it doesn't take over the whole thing. So we did a whole bunch of studies in these areas and have really worked all this out, and you can use probes for different phyla of microbiota, and basically in the mouse, I don't want to get into the details here, but there's basically 2 main phyla of microbiota, the bacteroidetes and the firmicutes, and the Citrobacter belongs to this thing. So this is the normal distribution of the phyla in a mouse gut. You then infect, the first thing you see is a major rearrangement in these things, and you start to see a little bit of red, that's the Citrobacter coming up. And by day 7, you can see there's more of it. But then by day 14, you're getting rid of the Citrobacter, and you're then starting to return back again. So there's obviously major shifts going on in the microbiota as you go through this. And again, I'm not going to go through the details of this, but really we found some profound things. And what we think the bacterium does is it goes in and it intentionally triggers inflammation. Now, that's a no-no from most bacterial pathogens, but we think it intentionally triggers inflammation that shifts the microbiota composition so it's more favorable for Citrobacter and its companions like E. coli to then proliferate. So it actually uses the inflammation to its advantage to change the microbiota so it's less competitive from Citrobacter point of view to shift it that way. Very radical concept. Now, we have a big meeting coming up here next Monday and Tuesday on microbiota, and there we will talk about the effects that we actually are starting to identify particular microbes that seem to be beneficial or harmful for Citrobacter to actually colonize these things. Okay, that's the microbiota. What are we going to do about it? We've found all these wonderful things with this neat biological system, but can we actually do anything useful? We're studying a disease. Can we do something to prevent this disease? So just to remind you, cows carry this thing. They secrete it. Cows don't get sick. They don't have the receptor for the toxin, but they contaminate water, or they by fecal contamination, or they contaminate ground beef when you make these meat products. So cows are the big problem. So most outbreaks come from cattle sources. And so I'm married to a pediatrician. I've been thinking for a long time of a childhood vaccine because this is a childhood disease mainly. And I was actually just out for a run one day out in the woods near the university, and the light came on that, well, we should maybe vaccinate the cows instead of the children, because that actually was where the source of the problem was. So again, a very unorthodox concept to develop a cow vaccine for a childhood disease. But that's what we worked with, because for a bunch of reasons, it's much easier to make an animal vaccine than a human vaccine. So we started to do this, and we returned to what we'd already learned about the type 3 secreted proteins I've told you about. And basically, we came up with the concept that can we vaccinate the cows with these proteins that allow the bacterium to adhere to the cell? And then when the cows would encounter this organism, instead of colonizing and then shedding large numbers, it would just come out the other end of the cow. It wouldn't cause these things, and we wouldn't have contamination. That was the concept. Bottom line, it worked beautifully. We can actually basically vaccinate these cows. And this is the very first experiment we did, where we basically took cows and we vaccinated them either with placebo or supernatants from the wild type. So here's our type 3 secreted proteins there. And then we basically fed them a bolus of 0157 and asked how many days did 0157 come out the other end of these cows. And if you don't vaccinate, they shed for about 5 days, whereas if vaccinated, they basically came in, went out the other end, that was it. Vaccine worked perfectly. Now, this vaccine has now been through several 100,000 cows. It works beautifully. It gets rid of the E. coli, takes it to a level that we normally see in the winter when you don't see this disease. The vaccine works. The vaccine is now being made. It's commercially available in Canada, and in the United States it's pending, as well as introduction in various places in Europe. Okay, we've done. We got the science, we got the vaccine, we're done, right? Wrong. Now, this is where interdisciplinary really comes into play. Question is, who's going to pay for this vaccine? Cows don't get sick, so a farmer says, why should I vaccinate my cow? My cows are just fine. And it's a very interesting problem. In Canada, they did a study that showed that basically vaccinated cows, if you vaccinate every cow in Canada, you would save 3 times the money it takes to treat this disease every year. Makes sense, but of course, there's different arms of the government, and so that concept is very difficult. In the United States, for example, they spent 3 years trying to figure out whether this should go to the FDA, which is human products, or the U.S. Department of Agriculture, which is animal products. This is an animal product for a human disease. They finally got it right and went with the agriculture, but that basically slowed us down there. So there's all these other things swirling around, and really you need policy change. And in the United States, what I think is going to tip the balance is basically the lawyers, because there's now lawyers that are suing meat producers if these kids get sick because there's a vaccine available and they're not using it. And I think that's going to tip the balance towards bringing in the vaccine. What does that have to do with science? There's your interdisciplinarity for you. So that brings me to the end, and I know it's been a quick survey through this, but I really think there's probably only 2 or 3 microbiologists in the audience, but I really want to stress that, when you tackle these problems, you tackle them from however you possibly can. So we have our pathogen here. I told you all about the neat, sophisticated disease they have and mechanisms they have. I told you about the host, the innate immune pathways, the T and B cells, all the host responses. And I told you also about the third player, the microbiota, that actually goes into playing this. And really, to understand this disease, you have to tackle all these aspects from all these different techniques. And then ultimately, I think you come up with a better definition of the problem, a better understanding of it, and in our case, even a solution to it. So this is just a general thank you to the many, many people who worked on this. This is an old picture from the lab, but obviously it's a thank you to the literally dozens of people that have contributed to these projects over the years. And with that, thank you very much.